Good evening. My name is Simon Leedham. I'm a Professor of Gastroenterology and a Wellcome Senior Clinical Research Fellow. It's also been my pleasure to be the Director of the Oxford Centre for Personalised Medicine over the last 4 years, which is a fantastic and progressive partnership between the Wellcome Trust Centre for Human Genetics and St Anne's College. We're an engagement centre that seeks to liaise and engage with a number of different demographic groups around the opportunities, the possibilities, the difficulties, and the challenges of integrating personalised medicine into modern-day healthcare. It's a great honour to be invited to participate in this St Anne's online course, with a fabulous initiative with some really fascinating multidisciplinary talks. And over the next half an hour or so, I hope to be able to take you through some of the issues and the challenges surrounding the introduction of personalised medicine into cancer therapy in the NHS. So I hope to be able to share my slides and get started so you can see what I'm talking about. So I hope that can be seen. I've entitled this talk, Hype or Hope? Personalised Cancer Medicine and the NHS. And this is on behalf of the Centre for Personalised Medicine from St Anne's and the Wellcome Trust Centre. So if we're talking about personalised or precision or genomic or stratified medicine, I think it's really rather important to understand what we're talking about. These different terms are used as synonyms really and fairly interchangeably. And what they all seem to mean is a type of medical care in which the treatment is customised for an individual patient. And from the Centre for Personalised Medicine perspective, we really think that the application of genomic technology to help tailor that healthcare has been the significant advance in the last few years. So these terms used interchangeably, I'll try and stick to personalised or precision, but all really mean the same thing. So to understand genomics and the impact genomics is having on healthcare, we really need to understand DNA, or deoxyribose nucleic acid, the so-called blueprint of life. DNA was, of course, the structure of DNA was, of course, identified in the 50s in Cambridge from Francis Crick and Jim Watson, here seen with this rather elegant helical structure. And here you can see the chemical structure of DNA, which is, in fact, a double helix loop with like a twisted ladder with rungs in the middle representing structures called base pairs. And it's these base pairs that are really critical in understanding how DNA functions. As I say, DNA is a little bit like a twisted ladder. The base pairs represent the rungs, and so they meet the double helix structure in the middle. And there are only 4 types of base pair, named for the first letter of their name: adenine, thymine, cytosine, and guanine, or A, T, C, and G. A always binds with T, C always binds with G, and it's the order of the sequence of these rungs on the ladder that people are talking about when they're talking about sequencing technology, when they're talking about genomics. It's understanding how these letters follow one after the other. So A, T, C, G, A, T, T, A, G, C. It's the order in which these base pairs follow on from each other that determines the instructions, the blueprint for life. Now, all this DNA comprises around 3 billion of these base pairs. The strands are super coiled up into structures called chromosomes, and we have 23 pairs of chromosomes in the vast majority of our adult cells. So how does this DNA code lead to cellular function? Well, it's all to do with providing the blueprint, the instructions for building the important functional molecules within a cell, and these are proteins. Like anything valuable, the DNA is kept locked up in the nucleus like a set of blueprints within a safe, the nucleus being the central area of the cell, and it never leaves the nucleus. It's super coiled up into these structures called chromosomes. When an instruction needs to be passed to form a new protein, a bit of the code is transcribed into a different molecule called RNA. The RNA then leaves the nucleus, here seen as the code passing out of the yellow nucleus into the blue cytoplasm of the cell, and it binds to a structure called a ribosome, which is, if you like, is a protein factory. There, the sequence of base pairs is interpreted, and according to the sequence, a different amino acid is dragged into the position, and those amino acids all then bind together to form a protein. Now, the way that an amino acid is structured really determines how a protein functions, and therefore the order in which amino acids go together is critical, and that order is determined by the sequence of the base pairs, the A, the T's, the C's, and the G's in the DNA molecule where the instructions started. So this is how this genetic code allows a cell to generate the worker molecules, the functional molecules of the cell, the proteins, and it's the proteins that determine how a cell then behaves and functions. So when we consider how genetics, how genomics has altered medicine, we're really starting to think about where are we with personalised medicine. And many people feel that personalised medicine is all hype. These are quotes that have actually been asked of me: what clinical benefit has genomics actually ever delivered? The margins of clinical improvement are small. People are concerned that the money spent on sequencing technology in genetics and genomics is not money well spent, and that this is rich country medicine that can't be afforded in developing economies and therefore leads to widening health disparities. And when I think about personalised medicine in terms of this, I like to use a model called the hype curve to illustrate this. Here we can see the hype curve, which has the visibility plotted on the y-axis and the maturity of a technology plotted on the x-axis. And you can see that every new technology starts with a technology trigger, rising with visibility rising rapidly to a peak of inflated expectation up here, before crashing downwards towards the so-called trough of disillusionment. This is then followed by gradual acclimatization towards a new technology, working towards the slope of enlightenment and eventually the plateau of productivity. So the question for this talk is, can we map personalized cancer medicine onto this hype curve? Well, when thinking about this, the technology trigger is relatively easy. It's to do with the sequencing of the human genome, which was completed in 2000, the year 2000, some 20 years ago. You can see a very youthful Tony Blair and Bill Clinton here announcing the breakthrough that was the publication of the human genome, mapping all 3 billion base pairs, incredible international effort leading to such a breakthrough. And I don't think Tony Blair was exaggerated when he said ever so often in the history of human endeavour, there comes a breakthrough that takes humankind across a new frontier into a new era. So that was the publication of the template of human genetics. It's really the template that we've continued to use ever since to refer to when we sequence any piece of DNA or RNA, we refer to the human genome, and that really was a breakthrough that's led to the development of many improved technologies over the last 20 years. So let's go to the peak of inflated expectation. And I think this is most nicely illustrated by the treatment of a condition called chronic myeloid leukemia. Leukemias are cancers of blood cells. This is a particularly unpleasant cancer of blood cells associated with a chromosome called the Philadelphia chromosome. What happens in chronic myeloid leukemia is that your chromosome 9 and your chromosome 22, remember chromosomes are supercoiled pieces of DNA, those two chromosomes break. A fragment of them breaks off, and as the DNA is repaired, because DNA can repair itself, it switches chromosomes so that we now have chromosome 9 with a fragment of chromosome 22 on it, and we also have an abnormal chromosome here, which is a fragment of chromosome 22, with the bottom bit being a fragment of chromosome 9. And this small chromosome, this altered chromosome 22, is called the Philadelphia chromosome. It's long been recognized to be present in patients with myeloid leukemia. When sequencing technology became available, people were able to sequence the Philadelphia chromosome and identify what the problem on it was that's causing the disease. And this disease is caused by the apposition of two genes that shouldn't be together. So here we have a gene called BCR, which is on chromosome 22, and a gene called ABL, which is on chromosome 9. And when these genes are separate, they are normal genes involved in the normal function and growth of blood cells. However, when those genes fuse, when the chromosomes break and the genes are fused together, they form a fusion protein or a fusion gene called BCR-ABL. And this unfortunately codes for a fusion protein, an abnormal protein, also called BCR-ABL. And here we can see a cartoon of that protein. Here we can see the abnormal protein called BCR-ABL formed by the fusion of two chromosomes together to generate a hybrid abnormal protein. This hybrid abnormal protein, unfortunately, loses its capacity to be turned on and off appropriately. So therefore, it continues to work all the time using an energy source called ATP within a small pocket of the protein to function. And it's really the continuous drive of this abnormal protein that leads to the cancer phenotype that we see in chronic myeloid leukemia. So when we were able to sequence that abnormal, that Philadelphia chromosome, we were able to see that two genes that normally aren't together were suddenly opposed and are generating a new and abnormal protein called BCR-ABL. When you can identify the protein, you can start to think about how to drug it, and that's what people did. They found a drug that was able to bind very specifically to the ATP pocket within this fusion protein and block it. And when you block the energy source of a protein, it can no longer function, and it blocks the abnormal continuous function of this fusion protein. And that drug was called Gleevec, or imatinib. And this was a remarkable therapy. So this was an archetypal therapy, one of the first of its kind, that was able to take a sequence identified genetic abnormality leading to an abnormally functioning protein, identify what that protein was doing, and therefore how to target it and drug it successfully. And this is the effect it had on therapy of this condition. This is a Kaplan-Meier curve, a survival curve, and we can see here that the survival in these dotted lines at the bottom here in 1975 from chronic myeloid leukemia, 5-year survival was about 20%. So 80% of patients, unfortunately, with chronic myeloid leukemia pre-1975 were dead within 5 years. And this is the effect that Gleevec therapy had on that survival curve. Here we can see the curve from 2001 to present, and now we can see that the 5-year survival curve for chronic myeloid leukemia patients who receive this drug, Gleevec, is something like 90%. So here we had a true targeted therapy based on a genetic condition, understanding what the genetics driving the condition was doing, and developing a brand new therapy to treat this disease very specifically and a very targeted way. So here I think we have the peak of inflated expectation based on the application of sequencing technology. And all of a sudden it appeared that different types of cancers were going to be vulnerable to different types of targeted therapies. However, unfortunately, the luster started to fade, and this is on the back of the discovery of tumor heterogeneity. Here you can see a picture of a nasty looking kidney cancer. And what these investigators here have done have taken multiple regions from across this kidney cancer, multiple biopsies or multiple samples from across this kidney cancer. And they then went on to sequence each of those sections. So they took a little piece across the tumor and sequenced across the tumor to understand how those areas were related to each other. And they were then able to draw pictures like this. This is a so-called phylogenetic tree to understand how the different regions were related to each other in terms of their genetics. So here we can see region 1 is closely related to region 2, but very distinct to region 4, and distinct separately and further to metastatic lesions or lesions that have spread from the original tumor. And what this work seems to show is that tumors, different areas of the same tumor, are much less closely related to each other than perhaps you might have predicted. And this suggests that the tumor evolves as it grows. Different pieces of the tumor, spatially separated, were genetically quite distinct from each other, which means that the tumor is evolving as it grows. And this is a problem for targeted therapies, because if you're trying to hit a target that continues to evolve, you're trying to hit a moving target. And that, of course, is much harder. And I think it's very valuable to draw the comparison here, as the investigators here did, between the phylogenetic trees they saw in these kidney cancers and the tree of life that Darwin speculated in his notebook when he was dreaming up his Origin of Species work. And this is because cancer is really a fundamental example of evolution in progress. Cancer evolves as it grows, and sadly, it evolves the capacity to avoid or compensate against our targeted therapies. And that's really where I'm moving on to now, the development of resistance to therapies. This is a paper based on malignant melanoma, a particularly unpleasant skin tumor. Here we can see a really very poorly patient with multiple melanomatous lumps all over his skin. This is skin cancer that's spread across throughout the body. This particular cancer, malignant melanoma, is driven by a gene called BRAF. This particular patient was one of the first patients in a trial to receive a BRAF inhibitor. So again, possible example of a targeted therapy, a genetic mutation leading to an abnormally functioning protein and the development of a drug specifically to target the abnormally functioning protein. And it had a remarkable effect in the first instance. Here you see the same patient having been treated with the BRAF inhibitor. The skin looks an awful lot better. The lumps have disappeared. You've been able to regain some of the weight lost during the cancer process. But unfortunately, very, very sadly, this patient's cancer developed resistance. So it became resistant to the BRAF inhibitor that it was being treated with, and the patient relapsed. And here you see the same patient, a sequence of pictures, now with newly developed resistant tumors. So the tumors that he got were suppressed by the drug, but unfortunately continued to evolve, developed resistance to the same drug, and then came back with a vengeance. And unfortunately, this was a fatal event for this poor patient. And therefore, when a doyen of cancer from the States, a chap called Bert Vogelstein, said, resistance is therefore a fait accompli. The time taken to recurrence is simply the interval required for a subclone to repopulate the lesion. This evolved and acquired resistance of cancers to targeted therapies seemed to sound the death knell for our hopes and dreams around improving solid cancer therapy. And I'd just like to point out how long this process all takes. Drug development, the average time for drug development is about 9 years. So new drug development takes a mean of 9 years to develop and costs around $2 billion when you take into account all the drugs that don't make it. And unfortunately, this, although it worked incredibly effectively in this patient, this BRAF inhibitor worked incredibly effectively in this patient, the time taken for the tumor to evolve and develop resistance was just 23 weeks. So an average of 9 years to develop a new drug, and a cancer is able to evolve and become resistant to that therapy within 6 months. So devastating, a devastating paper in 2011, reaching the plateau of disillusionment, I think, in terms of the potential for targeted therapy in cancer. So how have things progressed? Well, let's look at the cost of sequencing to start with. I mentioned that the first human genome was sequenced back in 2000. International effort, costs around $1 a base pair to sequence, and there are 3 billion base pairs. So the cost for the first human genome was about $3 billion. But since then, you can see that sequencing costs have fallen steadily and very sharply as technologies improved from about 2007 onwards, to the point where we are now. This is an old slide, as you can see, but the average cost of sequencing an entire human genome in today's market is probably about $1,000 per genome. So cost has fallen incredibly dramatically over the last 20 years as technologies have improved. And this really means that sequencing technology is becoming easier to use, it's becoming cheaper to use, and there is more and more data available. What does that mean? Well, it means that we have huge genomic resources now. So whenever a scientific paper is published, you have to publish the sequencing results that go with it. So that means that we start to build up these fantastic data repositories that allow people to further investigate the technology. Here you can see some work from something called the Tumor Genome Atlas, the TCGA. This is a huge number of different tumors, from pilocytic astrocytomas and brain tumors on the left here, all the way across to melanoma and squamous lung cancers on the right here. And this is the range of mutations seen in different types of tumors. So you have very few mutations seen in these brain cancers, but much log scales larger numbers of mutations seen in melanoma and lung cancer. And it's perhaps because these are very high in mutations because they're caused by an environmental exposure, which is sunlight or smoking, respectively. And what does this range of of mutation burden mean? Well, as I mentioned before, genes give rise to proteins, or genes code for proteins. So if you have a large number of mistakes or mutations in your DNA, you give rise to a large number of different or abnormal proteins. And that's something that we've recently been using as a vulnerability for cancers. So tumors with a lot of mutation in it generate a lot of new proteins. Those new proteins can be recognized by the immune system because our immune system recognizes abnormal protein, and therefore immune cells can infiltrate into cancers and they have the capacity to kill cancer cells. So wonderful work on drugs that help stop the tumor from invading these immune cells led to the Nobel Prize a couple of years ago, so-called immunotherapy. And we've seen remarkable results in cancers with many mutations, such as melanoma and lung, from immunotherapy, which is really harnessing the body's own internal weapons to fight a cancer and not allowing the cancer to escape that fight. So where's the future of personalized cancer therapy sit? Well, I think it really, this is the sort of dream, the ideal of personalized therapy. It relates to this sort of grey population here that receive a grey pill, and some patients will, this is the one-size-fits-all approach. Some patients will have a response and unfortunately no toxicity. Other patients will have a response but side effects. Some patients will have no response at all but no toxicity. And some patients, unfortunately, will not only have no response, but they'll get toxic side effects from drugs. So this one-size-fits-all approach leads to benefit in some patients, but zero benefit in other patients. The dream of personalized medicine is that we take that grey, homogeneous population and we stratify them somehow. We stratify them with genetic technology, we stratify them with sequencing so that we understand the disease subtype in each of the patients. So we turn our grey population into a red population, a blue population, a yellow population, a green population by this molecular stratification. And then we can apply drugs that have been specifically designed to target the genetic vulnerabilities that we identify in each particular disease subtype. So we have a red pill for red patients, a yellow pill for yellow patients, a green pill for green patients, and so on. Therefore, that the right medicine is given to the right patient at the right time. So where does genomics sit in the modern day NHS? Well, I think I'm proud to say that it sits front and center. The UK has led the way in terms of integration of genomic technology into its healthcare system. Of course, we have the perfect healthcare system to do that, and we have a centralized national health service. The annual report of the Chief Medical Officer back in 2016, it was called Generation Genome and was all about the integration of genetic technology, with Dame Sally Davies saying, now we need to welcome the genomic era and deliver the genomic dream. And we've had a fantastic program since 2012 called Genomics England, which has sequenced something like 100,000 whole genome sequences across a range of diseases. And has really been very integral and important in establishing genetic and genomic technology within the NHS. There are a number of issues with this, of course. Although, of course, many people will be able to see the benefit of sequencing technology and science has brought to the fight against coronavirus. Clearly, the costs of sequencing technology are still prohibitive. They're still extremely expensive, even though they are, of course, falling steadily, as I showed you before. I think as technological improvements and capacity builds over the years to come, there will be scalable availability, and there's been movements following on the back of the Genomics England program to introduce genomic medicine across the country, which has been a hugely valuable infrastructural effort. I think the molecular stratification should definitely inform the next generation of clinical trials, and there's lots of collaboration with industry to make sure that patients are properly stratified to the right drugs to minimize toxicity and increase benefits. And I think leading the way internationally helps UK PLC. It's certainly something we're recognized for in our science across the world. And then a hypothetical question here is, can we afford not to? Can we afford not to integrate the advances in the sort of scientific technology to improve patient care? And I think really we have to try and find the money to do this, because this could lead to improvements in care and the reduction of side effects for patients in the future. What about clinical interpretation? Well, I mean, there is no question that the exponential increases in technological capacity in terms of the scientific advances have greatly outstripped our ability to understand these findings in light of clinical disease. So clinical disease can't really keep pace with the scientific advances, and we need to be including understanding of genetics, genomics as part of medical student training programs. We need the doctors of the future to be engaging with genetics and genomics, because I hope that it'll be a powerful tool that they'll be able to use in their clinical practice for the future. Genomics England has established specific training programs to help do this, and engagement forums like the Oxford Centre for Personalised Medicine look to push this agenda wherever we can, even if it is just at local scale. It's very important that we keep public enthusiasm for this up, because there's a bewildering and exponential increase in knowledge that it's very hard to keep abreast of. There are often stories in the media that are not necessarily very favourable, such as the postcode lottery drug stories. And as I mentioned before, the pace of change is glacial in comparison to the scientific technology when we're thinking about translational improvements. And what I mean by that is that if an average drug takes 9 years to develop, then actually the medical advance may be 9 years before a drug can be delivered to help treat a disease, a new disease that's been identified, or a new problem that's been identified. Where genetic or genomic technology is discussed, there is always the query or the fear of eugenics, and of course hype is something that we've been discussing all the way through. The pace of change can be disappointing for people, and people therefore start to think that personalised medicine is all bluster and no actual material. And then finally, a really important point to consider when trying to integrate genetics and genomics into healthcare is data use and protection. A sequencing experiment produces a huge amount of data, gigabytes and gigabytes of data for a single genome, and that data needs to be stored, analysed, and kept safe. There is a lot of suspicion and caution over data protection and confidentiality, and there, I think, is inherent embedded mistrust from the public of the use of NHS data for the benefit of private companies, particularly with insurance, but also to some degree around pharma. Even though it's the pharma companies that do the vast amount of new drug development. I think there was a fantastic review by Sir John Bell recently that suggested that capitalisation on the unique NHS data sets should be encouraged, but perhaps to use the Department of Health as a benefactor, and I like that idea. So to summarise, where are we? Where is personalised cancer medicine on the hype curve? Here is the hype curve again, and if I plot where I think we are with this, I think we can see that the technology trigger was sequencing the human genome, and the peak of inflated expectation was met soon afterwards with the successes of the early targeted therapies, such as Gleevec for chronic myeloid leukemia, dealing with the fusion protein I discussed, the BCR-ABL protein. Unfortunately, the understanding of tumour heterogeneity and clonal evolution led to issues with targeted therapies. And then when we saw short remissions with targeted therapies, with the rapid development of evolutionary driven resistance, there was a lot of fear that genetics and genomics weren't going to be able to deliver on the promise of new cancer targeted therapies. So where are we now? I think we're around here. I mean, this is an arrow picked out of the ether, but I think we are definitely working towards the integration of genetic and genomic technology to help tailor healthcare. This is the slope of enlightenment heading towards the plateau of productivity. Many new advances in our understanding of cancer biology have been developed on the back of sequencing technology, of genetic and genomic technology. We have the opportunity, I think, to be able to use understanding to develop new medicines. There will be an inevitable delay as those new medicines are tested and checked and make it through the very, very rigorous rigorous clinical trial proformas. But I think if coronavirus, if anything, has shown us, the pace of scientific advances has rapidly increased with the application of new scientific technology, and perhaps the pace of drug development could develop just as quickly if academia and pharma can work together. So I'd really like to thank you all very much for your attention. I can be contacted through the Centre for Personalised Medicine if you have any questions. Unfortunately, this isn't a live broadcast, but I'm always very happy to talk to people around precision or personalised medicine. These are the contact points for the Centre for Personalised Medicine. Please do visit our webpage, have a look. There are a huge range of videos there around precision and personalised medicine on a wide range of multidisciplinary topics. The Oxford CPM, as I say, is brought to you by the Wellcome Trust Centre for Human Genetics and St Anne's College. Fantastic and very progressive partnership. It's funded predominantly by the Stanley Home Medical Development Foundation, who have supplied funding for the CPM for the last 7 years, and we have a fabulous relationship with our friends and partners in Macau, Hong Kong, and mainland China. And we have a contribution too from the Wellcome Trust. So I'd really like to thank you all very much for your attention this evening. As I say, if there are any questions, please do contact me, or if there are any topics that people would like to be discussed or to discuss in a Centre for Personalised Medicine event, then I'm always very interested to listen to new ideas. We are particularly fond of interdisciplinary and multidisciplinary initiatives, so please do drop me a line if you're interested in this. Thank you all very much for your attention, and I wish you all a very pleasant evening, and please stay safe in these troubled times.